From the book? Which one? Immunology you have not read. I have taken 2 classes. Have you read the notes? None of you have read. That will be very difficult, I am telling you. You cannot understand. It is difficult, I am telling you. The subject is as such difficult. And deliberately, instead of taking 1 class, I am taking 3 classes. I have already covered 2. This is the third class. If you do not read at all, even after discussing everything, and if you are noting down, if you are not going through the notes also, how will you have a link about this class, today's class? Because today's class will be basically what I have told in the previous 2 classes, and in addition we are giving some new informations. I am going slowly, very slowly, as I told you earlier. And now from onwards I will start telling 2 or 3 wrong things deliberately. And I want to see whether you are at all listening or not. If sitting idle will serve no purpose. If you can write, if you do not read, then how will you remember? It is not that easy. I told you, I explained the differences of 3 types: innate, adaptive, primary, secondary, active, passive. Questions will be asked basically on these 3 charts. And to understand that thing, I have explained in detail. Again today I will cover. See, last time I told you this, can you remember this picture? Some of you are fond of taking photographs. Why I am not allowing? I am telling you, you have to go there and again in the hostel, and then you have to see the small screen. You have to again note down. That will take a much longer time rather than noting it directly from here, from the big screen, and you can at the same time start understanding. So this part I have told you is the T cell receptor. There is one receptor, and in contrast B cell receptor is having one receptor and the same immunoglobulin is produced. This is in continuation of the previous class. I am showing you this just to remember. Cell-mediated clone expansion, clone selection, expansion and creation of memory cells I have told. Do you remember? And I told you about this difference between these 2, between T cell and B cell. B cell produces antibody instead and memory cells. Instead, T cell also produces, instead of antibody, they produces 3 different types of cells. One is T cytotoxic, one is T helper, another is T regulatory. And I stopped here about this 4, only single function of each type of cell, so that you memorize at least this much. And naive T cell I told you, or naive B cell, which has not come in contact with any antigen so far. So that is called naive T cell. Now this is part 3, this is last part I will cover. I may take a little longer time because I am starting this class a little late. So note down about from these 3 things, what are the functions of the cell-mediated immunity. See, many times we cannot kill the bacteria, viruses, even parasites. So these parasites, viruses, bacteria are engulfed by the phagocytosed. Phagocytic cells they are killed by the T cells. So immunity against many intracellular bacteria basically. Most important is Mycobacterium tuberculosis, and almost all viral infections are killed by the T lymphocytes. Rejection of allograft. If somebody is bone marrow, somebody's kidney, somebody's heart is taken and transplanted into somebody else, and where there is mismatch, some amount of mismatch, and it is going to happen if it is not an identical HLA haplotype matching a sibling, it is likely, and that is caused by T cells. Tumor cell destruction, this is another important function, because if cancer cells, tumor cells are not destroyed in our system, they will start growing. So T cell destroys that thing. Helper function, as I told you, T helper cell, I told you last time that it helps in the production of antibody. So if T helper cells are destroyed, and which happens in AIDS, antibody production will be hampered or reduced. In addition, it also generates cytotoxic T cell. So T helper cells are producing 2 things. One is helping in the production of antibody and also production of the cytotoxic T cells and suppressor cells. I told you about regulatory T cells, so that regulate and it down regulates both T and B cells also. At times it causes suppressor function also. Regulatory means it can regulate, it can upregulate, it can downregulate. One just example, suppressor function that is also caused by T cells. Now T helper cells, I told you earlier, only two more extra point you raise. It releases cytokines. So it can come as a short note. Cytokines I am telling you in a short while. So it helps in the antibody production, I have told you. Antibody B cell class switching I have told in the primary immune response. There is IgM, and in the secondary response other 4 types are produced, that is IgG, IgA, IgD and IgE, and predominantly IgG. And this class switching is the function of the T helper cells. And you write down this one, activation and growth of cytotoxic T cell, just I told you. Only these 3-4 underlined sentences you remember, the rest are difficult, you may not, need not memorize. So specifically T helper cells has these functions. Cytokines, I will only tell one slide, you note down this thing. If a short note comes, at least you write down this one. Many things have to be told, at least half an hour time will be required to cover cytokines. But cytokines, what are cytokines? Basically small proteins. They are signaling molecules. See, you are admitted in this hospital, you are staying in the hostel. So how this signaling was transferred? You came, you went to the administrative building, your name was registered, you were allotted in a seat. Some sort of signaling is there. That's why you are staying in that room, in that hostel. Some signaling is there. Similarly, today you are attending here, some signaling is there. So cell to cell signaling must be there. Otherwise, cell will not function properly. So that is done by the cytokine cells. So important function is cell signaling, and they also regulate the growth and activity of other immune cells, and it's a soluble mediator of inflammations. These are cytokines. These you will be, I have been told in detail later. Write down this thing, interleukin. Interleukins are ILO. There are many interleukins. More than 20 different types of interleukins are there. So only one line you write down. It regulates the innate and adaptive immune response. There are many functions. Just for your own information, minimum information, you write down this only one single point. Rest you can definitely get from the textbooks, and as much as you can memorize, it will be helpful. Interferons, they are basically are antivirals. And you write down this thing, interferons are designated as IFN. IFN alpha, beta and gamma, they are antiviral. Tumor necrosis factor, mediators of inflammation. These you will read in detail about in the physiology class or maybe in the pathology class also in future. A granulocyte colony stimulating factor, they help in differentiation and maturation of granulocytes in the bone marrow. And in cancer, after giving chemotherapy, destruction of cancer cells, some of the bone marrow cells are also destroyed. That's why to revive those cells, the GM-CSF is given routinely almost after chemotherapy, and that you will be using also. At times you will be giving granulocyte colony stimulating factor, or you will be giving granulocyte macrophage stimulating factors by injection, so that the bone marrow cells which were destroyed, they are also regenerated. So usually because of the destruction of the cells, the count of the cells goes down after giving chemotherapy. Cells level come down, HB levels comes down also. Now there is a cycle after chemotherapy. I am telling you why I am explaining. Today you have given the chemotherapy completed after 4 or 5 days cycle. Now after 3 weeks you will be given another chemotherapy. So during this period before chemotherapy, these counts are taken. Once chemotherapy is given, that destroys cancer cells. At the same time, that produces the highly proliferating cells of our body. One important thing is bone marrow cells. So, the bone marrow cells, they are also destroyed, they are highly proliferating. So, now to regenerate those cells, these granulocyte stimulating factor, choroidal stimulating factor is given by injection, so that the count gets increased, HB level gets increased by the time the patient is almost ready for the next chemotherapy. So, this is the clinical application. So, macrophage migratory inhibitor factor, that is another function of cytokines that we have to regulate somehow, and C-reactive proteins. Now, do you get my point? So, these are the actual all are clinically relevant things, and you will be using also interference, you will be using clinically. So, just to summarize, immunity we have divided into two parts, one is innate, one is adaptive. Innate is from birth. Adaptive, there are two types, one is natural, means we have got from something from the mother, that is passive, and we have actively developed immunity, and artificial. Passively somebody is suffering from some disease, a gas gangrene or tetanus, we have given immunoglobulin. So, passively we have given the actual immunization. So, that is a short lived and that will be destroyed after some time. And in contrast, we get an active immunization which all of you have taken, polio, DPT, tetanus, measles. So, these are all long lasting, almost lifelong you will get this immunity. Tetanus you will be required to take booster doses after some years, 5 years, 10 years. Okay. Rest of the vaccines you are almost immune, and those diseases are common in the childhood. So, you are least likely to suffer from those diseases. So, antibody, now today's class is thrust is basically antibody. I will tell in detail, just write down, I told you earlier. Hold on, you don't write down. What I have to, you have to write, I will tell you. I told you about epitope, that is antigenic determinant. Whole part of the antigen is not determining. Suppose how you are being recognized, you are recognized by your face or through retinal images or through fingerprints. So, these are basically the determinants of your identity. So, the whole antigen does not get involved into the antigen production, antibody production. So, the antigenic determinants are actually involved in antibody production, and antibodies are produced against that region. So, that region of the antigen is, which is antigenic determinant, is called the epitope, and that which is produced by the antibody is called paratope. So, as I told you earlier, if when you are interacting with somebody, either you are handshaking, that is the minimum information. So, you are communicating with this thing, binding with here, or at the best you are hugging. So, it depends how do you interact with the antigen antibody. So, that is determinant factor of the antibody. How many antigens are there in that particular parasite or particular virus or particular bacteria. If there are multiple, so it has to be just like hugged, or you have to embrace. Or if it is a single epitope, you will handshake simply, so that there will be touching. Do you get it? So, paratope is basically part of the antibody and epitope is a part of the antigen. Now, this definition you can write down, this one, better definition is there. So, as I told you, the B cells produce antibody, but B cells as such does not produce. It gets converted into plasma cells. So, what is antibody? Antibody, again the protein classification we have said that there are some conjugated proteins. Proteins get conjugated with something else. Here antibody is not a pure protein, it is a glycoprotein. So, carbohydrate part is there. It is found by the immune system, that is plasma cells, and these plasma cells are differentiated B lymphocytes or B cells, whatever you say. Specifically it is created by the plasma cells, loosely we can say B cell. In response to antigenic stimulus, otherwise it will not produce. B cells are circulating, but all are not producing antibody, and recognizes a particular epitope of an antigen. You understand what I am telling right now? And bind specifically to it. Without that thing it will not bind, it will bind specifically to that determinant. This is the simplest definition you can remember. One good definition I am telling you, this I will explain. You note down this thing. And in a little while I'll explain this thing. B cell produced molecules encoded by genes. That is a protein molecule, therefore that will be encoded by the genes that rearrange during B cell development, because B cell develops into plasma cells. So that genetic rearrangement is there. I will show you within 5 minutes how these genetic cells are arranged. Of immunoglobulin heavy chain and a light chain. দুটো জিনের কম্বিনেশন হয়, জিন রিঅ্যারেঞ্জমেন্ট হয়, দ্যাট টুগেদার ফ্রম সেন্ট্রাল কম্পোনেন্ট অফ বি সেল রিসেপ্টর। এই বি সেল বলেছিলাম একটা বি সেল একটা অ্যান্টিবডি সিক্রিট করে আর একটা অ্যান্টিবডি সেম অ্যান্টিবডিটা এর উপরে রেখে থাকে। তাকাও, সেন্ট্রাল কম্পোনেন্ট বি সেল রিসেপ্টর ফর অ্যান্টিজেন, এই বি সেল রিসেপ্টর ফর অ্যান্টিজেন, এখানে অ্যান্টিজেন এসে বাইন্ড করবে। or secreted as molecules in the plasma or body fluids. অ্যান্টিবডি সিক্রেট করছে, হয় প্লাজমাতে করবে নাহলে বডি ফ্লুইডে করবে, মিউকোসাল ইমিউনিটি তৈরি করে, মিউকোসাল মেমব্রেন তৈরি করে। This is a better definition. If you say this thing, this type of things, then that means you understand. Now this gene arrangement I am telling you, I will tell you. So before that, this I have discussed in detail last time also, again I will tell, because this is most important to try to understand and stop me anywhere. এই ডাস্টারটা কোথায় গো? ডাস্টারটা রেজিনা, একটু ডাস্টারটা দেবো তো। দেখো ভালো করে, এটা জাস্ট রিমেম্বার। এই স্ট্রাকচারটা বই সব বইতে পেয়ে যাবে, কোনো ঝঞ্ঝাট বোঝার চেষ্টা করে। এই জায়গায় তোমরা এখানে কাছে আছো, এদিকে ডাস্টার আছে, এই বইয়ের ছবিটা সব জায়গাতে পাবে, শুধু এখন এখানটা একটু বুঝে নাও, ঠিক আছে? এর দুটো হেভি চেন থাকে। হেভি চেন কেন বলছি, এই দেখো স্ট্রাকচারটা অনেকটা বড়, আর দুটো লাইট চেন থাকে, ঠিক আছে? এই দুটো চেন ডাইসালফাইড বন্ড দিয়ে অ্যাটাচ করা থাকে, দুটো ডাইসালফাইড বন্ড থাকে এখানে, ঠিক আছে? এখানেও ডাইসালফাইড বন্ড থাকে, এখানেও ডাইসালফাইড বন্ড থাকে, ইন্টারচেঞ্জ বাইন্ড করবে, ধরে রাখতে হবে তো দুটো। এইটা হেভি চেন, এইটা লাইট চেন, এই দুটোর কানেক্ট করতে হবে তো, কোনোভাবে কানেক্ট ফিক্স করতে হবে তো। আইদার কি করতে পারে, নন কোভ্যালেন্ট বন্ড দিয়ে করা যেতে পারে, ইলেকট্রোস্ট্যাটিক অ্যাট্রাকশন, হাইড্রোফোবিক বা হাইড্রোজেন বন্ডিং দিয়ে করা যেতে পারে, বাট দ্যাট উইল নট বি স্টেবল স্ট্রাকচার। সো ডাইসালফাইড বন্ড ইজ এ স্টেবল স্ট্রাকচার। সো ডাইসালফাইড বন্ড দিয়ে এটা অ্যাটাচ করা থাকে। তাহলে এইটা যদি হয়ে থাকে তাহলে ইন্টারচেন, মানে এইটার সাথে এটা একটা সেপারেট চেন, এটা একটা সেপারেট চেন। সো দেয়ারফর দ্য টার্ম ইজ ইন্টারচেন। বোঝা গেল? তো যদি দুটো ডিফারেন্ট জিনিস হয় দ্যাট ইজ ইন্টার, আর যদি সেম জিনিস হয়ে থাকে তাহলে সেটা ইন্ট্রা। এই জায়গাটা হয়েছে এই যে হেভি চেনটা, এই হেভি চেনটা এইখানে একটা ডাইসালফাইড বন্ড আছে, সো দিস ইজ ইন্ট্রাচেন, এটা ইন্ট্রা। তাহলে ইন্টারচেন ডাইসালফাইড বন্ড দিয়ে এটা অ্যাটাচ করা থাকে হেভি চেন আর লাইট চেন। অ্যাট দ্য সেম টাইম এই যে দুটো হেভি চেন আছে, তো দুটো হেভি চেনকেও অ্যাটাচ করতে হবে। সো ইউ রিকোয়ার অলসো ডাইসালফাইড বন্ড হেয়ার, and there will be different structures of the immunoglobulin. Because of that thing, there might be intrachain disulfide bond also. So basically, this heavy chain are of 5 category. একটা হচ্ছে IgM, I told you earlier, একটা IgG, এইটা প্রাইমারি রেসপন্সে তৈরি হয়, আর বাকিগুলো সব সেকেন্ডারি। IgG, IgD, IgE, IgG, এম মিউ আরটা কি আছে আইজিএ, সরি আইজিএ। এই যে এম চেঞ্জগুলো হচ্ছে 5টা ডিফারেন্ট টাইপস অফ হেভি চেন। এইটাতে যে হেভি চেনটা থাকে তার নাম হচ্ছে মিউ। এইটা যে জিটাতে যেটা থাকে সেটা হচ্ছে গামা, ডিটাতে যেটা থাকে সেটা ডেল্টা, ইটাতে যেটা থাকে সেটার নাম এপসিলন, আর এটা যেটা সেটা আলফা। ঠিক আছে? তো আইজি এ জি যেটা থাকে, আইজি জি, আইজি ডি, আইজি ই, এই তিনটেরই স্ট্রাকচার এইরকম, মানে দ্যাট ইজ এ মোনোমার। Only there is a difference in the antigen antibody and amino acid sequence composition, and that's why their molecular weights are different. Okay? Only IgA তে দুটো মোনোমেরিক ইউনিট থাকে, যেটা একটা চেন দিয়ে জয়েন করা থাকে, সেটার নাম হচ্ছে জে চেন। সো IgG, IgD, Ig is a monomer, IgA is a dimer, আর IgA 5টা লাগানো থাকে এরকম। So therefore it is called a pentamer. এখানে একটা জয়েন করা থাকে চেন দিয়ে। তাহলে সাপোজ মনে করা যদি if it is the molecular weight of this one is x, so this will be basically 2x minimum, around 2x প্লাস এই যে চেন যেটা বাইন্ড করছে সেটা আরেকটা হবে y হবে, তাই তো? So it will be somewhat higher. Similarly here it will be again 5x প্লাস ওই যে চেনটার জন্য যেটা হবে সেটা y। বোঝা গেল? This I will discuss in the slides, you will note down. Right now you understand. So this heavy chain I am telling you again, এইটা 5টা টাইপের হয়, এইটা 2টো টাইপের হয়, ওয়ান ইজ কাপ্পা, ওয়ান ইজ ল্যামডা। একটা ইমিউনোগ্লোবিন মলিকুলে আইদার কাপ্পা চেন থাকবে কিংবা আইদার ল্যামডা চেন থাকবে, there will be no mixture. Normally in our human body almost 2/3 থাকে কাপ্পাটা আর 1/3 থাকে ল্যামডাটা, বা 60% বা 40% মনে রাখতে পারো। মানে যত ইমিউনোগ্লোবিনটা সমস্ত স্টাডি করা হয়, এর 4টে পার্ট আছে, এই পার্ট, এই পার্ট, আর এই ওপরে যে একটা পার্ট আছে সেটার নাম হচ্ছে ভেরিয়েবল পার্ট, আর বাকি যে জায়গাটা আছে সেইটা কনস্ট্যান্ট পার্ট আছে। Only variable part binds with the antibody. Only variable parts binds with the antibody. That's why it is called FAB. এই রিজিয়নটা। হেভি চেনেরও একটা পার্ট থাকে, একটা পার্ট হচ্ছে গিয়ে ভেরিয়েবল পার্ট, একটা পার্ট হচ্ছে কনস্ট্যান্ট পার্ট। লাইট চেনের, সরি, লাইট চেনের একটা পার্ট ভেরিয়েবল, একটা পার্ট কনস্ট্যান্ট। সিমিলারলি হেভি চেনেরও একটা পার্ট ভেরিয়েবল, একটা পার্ট কনস্ট্যান্ট। এই পার্টটুকু গোটাটাই কনস্ট্যান্ট থাকবে হেভি চেনের। This part only binds with the antibody. That is the reason it is called FAB portion. আর বাকি পার্ট যেটা রয়েছে এইখানটাকে বলে FC portion. That is crystallizable, fragment crystallizable. fragment antibody binding, fragment crystallizable. এইখানে কমপ্লিমেন্ট বাইন্ড করে, বোঝা গেল ব্যাপারটা? এটা বুঝে নাও, বাকিগুলো তো স্লাইডে দেখিয়ে দেবো, বইয়ে সমস্ত লেখা পাবে, বুঝে নাও জিনিসটা, ব্যাপারটা। এখানে একটা জিনিস জিজ্ঞেস করা হয় এক্সামিনেশনে, অল অফ ইউ আর বেঙ্গলি, ইজ দেয়ার এনিওয়ান নন বেঙ্গলি? No, you are not understanding, you raise your hand if I by chance, because most of the time I am speaking in Bengali. You try to understand. If you do not understand, or if any non-Bengali is present, please let me raise your hand. So, very often the questions are asked: pepsin digestion and papain digestion. What is the end result? Actually, I told you this structure is like this. If I digest with pepsin, it will cut here. There is a disulfide bond, it will be towards carboxy terminal, it will bind. So you will get a two fragment, one is two, এইখান থেকে যদি কেটে দিই আমি, জাস্ট কেটে দিলাম, তাহলে এইখানে দেখো একটা দুটো এফ এ বি টু, এই দেখো দুটো একসাথে বাইন্ড হলো, দুটো পিসে তৈরি হবে, দুটো ইফেক্ট। পেপসিন যদি ক্লিপ করা হয়, পেপসিন যদি অ্যাক্ট করে, পেপসিন এরকম জায়গাতে কাজ করে, it will be towards carboxy terminal, because amino terminal is this side. So it will make two pieces. One piece will be FAB2 and one piece will be FC. আর প্যাপেন দিয়ে যদি আমরা ডাইজেশন করি, তাহলে যে ঘটনাটা ঘটবে সেটা এরকম হবে। এই দুটো এফএবি, টু এফএবি ফ্র্যাগমেন্ট তৈরি হয়ে যাবে, বুঝেছ? এই দুটো ফ্র্যাগমেন্ট আলাদা তৈরি হয়ে গেল। এইটা একটা, দুটো এফএবি তৈরি হয়ে গেলে একটা এফসি তৈরি হয়ে যাবে। প্যাপেন, পাপা, পিএ পিএ, তার মানে দুটো তৈরি হবে ওখানে, দুটো এফএবি ফ্র্যাগমেন্ট তৈরি হবে, একটা এফসি ফ্র্যাগমেন্ট তৈরি হবে। এখানে একটা এফএবি ফ্র্যাগমেন্ট তৈরি হবে, কিন্তু দুটো বাইন্ড করা থাকবে। এখানে দুটো ফ্র্যাগমেন্ট, ওটা দুটো পেপসিন, মনে রাখবে দুটো, দুটো ফ্র্যাগমেন্ট, প্যাপ এন তিনটে ফ্র্যাগমেন্ট, পাপা দুটো সেম ফ্র্যাগমেন্ট তৈরি পাবে। Why is it is important? Why should I know this thing? Because if we inject the whole antibody to anything, anyone, because most of the experiments are done from the animal experiments. So suppose we know that this part binds with the antibody, antigen. So how to know different functions of the different part of the antibody? For that we have to break it down and we have to see which structure is binding. Suppose I am telling you complement is binding here. So if I take the whole antibody and if I allow it to bind with the complement, I will not know where it is binding. Now if I separate these two parts, if I allow it, the reaction to occur, only these parts which is having the FC portion will bind to that thing. Similarly, the FAB2. So these are all required for experimental purposes. And another function is that when the molecule is in the Y form, just like this one, it can bind very tightly and there is a flexibility. This Y region can flex, bind, and it can tightly accommodate also. If there is two identical antigens and the antigens are bigger, then it can tightly bind because the two hands can come together. If I make a fragment like this, It will not be able to do that thing. Now, why this is important? Because clinically, if you give the molecule, there is a chance that we can, or human being can develop antibody against that antigen because this is a foreign protein. Now, for some situations, since this is binding with the antigen or antigen, so we can use this part. Suppose there is a patient has been given a high amount of drugs, or the drug level in the blood is high. Now, how to remove that thing? Suppose digoxin is given for the cardiac patients. Now, digoxin overdose is there. Now, digoxin has to be removed from the blood. So, this FAB fraction, if you allow anti-digoxin antibody, and if you inject that thing, the extra amount of digoxin will bind to it. So, digoxin will not be available, that drug. Similarly, if there is a snake venom, viper bite, venom is there in the system, it is causing, we have to give antiserum can cause reaction. Now, instead of that thing, if we give against the same toxin, if you give the FAB fragment only, so that will bind and that will neutralize. There is no need of giving the whole antibody. Do you get my point? So, this is having one part is research part to understand the functions of different part of the antibody molecule, and one is therapeutic application. Similarly, cancers, anti-cancer drug can be bound, tagged to it, and it can be injected. In the eye, it is given, and you will be injecting intravitreal injections, anti-endothelial growth factor injections will be there, and these are tagged to FAB fragment. Do you get my point? The clinical application, these are not written in your book. Because why should I digest? What is the purpose of knowing papain digestion and pepsin digestion by you people? It's purely biochemistry. Why to get two fragments? And what will you use it? What will you do that thing? So now you note down, there I told you antibody binding site, it is clear. Now these are three parts. This is constant part, this is variable part. Now, disulfide bond I have told you, antigen binding site I have told you. This part clear? Okay. So again I am telling you the basic task, two same heavy chain, not all other. Two same light chain. And either kappa or lambda. A variable region is this top part, remember. And again in the variable region there is one hyper variable region, and that is determines specificity. Write down one part in the variable region, there is a hyper variable region. I will take a little longer time because I need to explain this thing. So function of IgG, only you write down this thing, this red parts. What is agglutination? Have you seen anybody blood group testing? Blood group testing you have seen? Okay, fine. So basically here there are two binding sites. Suppose I am just writing Y, okay, just for simplification purpose. So it will bind one antigen here, one antigen here. Now normally this type of antigen antibody reaction causes precipitation, not agglutination. Agglutination occurs when there is a visible reaction. Particulate matter এর সাথে যদি কোনো একটা particulate matter এর সাথে লাগানো থাকে তাহলে রিঅ্যাকশনটা ভিজিবল হয়ে যাবে। So just write down, one is immunoglobulin formation, binding, immobilization, কারণ কি অ্যান্টিজেনটা অ্যান্টিবডির সাথে বাইন্ড করবে। Usually pathogenic organism or it is a toxin. So that is an agglutination. Agglutination I will tell you, IgM is a better anti-agglutinating effect. Why I will explain. Coating the surface of the pathogen, because the pathogen is there, so this surface is coated by this IgG type of antibody, so there is a it will be helpful for the phagocytes to engulf it. So that is called opsonization. Means coating, something we are giving a rough surface and something we are coating it so that the surface becomes smooth, just like that. And there is one thing called antibody dependent cell-mediated cytotoxicity. That is the basic fundamental concept of opsonization. activation of classical pathway and complement system. There is another thing you just note down. Neutralization of toxin, as I told you, because antigen toxin will bind, against that toxin antibody was dealt. Snake venom is a toxin, so we have given an antiserum, so antiserum will bind to that thing, or we can give FAB also. So that will neutralize that toxin, it will bind. Antibody dependent cell mediated cytotoxicity, that's what I told you. It's another one chapter I have deliberately missed it at the end. If you are interested, I can explain it to you. And you say type 2 and type 3 hypersensitivity reaction, and this is beyond your scope. So at least you write down agglutination, opsonization, neutralization. One thing you remember, at least neutralization of toxin and opsonization. These two parts at least you remember. Agglutination you can write down together, or immobilization and binding together. You need not write agglutination, write down immobilization and binding together. This part I have explained in detail. This is a normal structure, immunoglobulin IgG, Ig, IgA. This is dimer, IgA. These are all present in the mucous secretions. So they give surface mucosal immunity. Like we are having in the nose, inside the nasal epithelium, we can get GI tract, genital tract, urogenital tract. But there is epithelium, there IgA is present, particularly gastrointestinal system. This is pentamer. So pentamer is having a high agglutinating activity because it can bind 10 antigens, and it's called visible clumping. And what you see in the RBC, that blood grouping, basically RBC is a particulate matter, it is particle. On the surface the antigens are there, and if you give an antibody, that antibody will react with the antigens present on the surface, and IgM type of antibody is usually normally used. So many RBC will be clumped together, so at least 10 RBC will be clumped, bound to the IgM molecule, and it will cause a visible clumping. That visible clumping is called agglutination. Simple antigen and antibody reaction, you may not get any visible clumping. That needs to be centrifuged so that to get a precipitate. That is the difference between agglutination and precipitation. Agglutination requires particulate matter, and IgM is a more suitable antibody rather than IgG. With IgG we can use some lattice particle, we can use any particulate matter attached to that IgG molecule, we can get a clumping also. Affinity, avidity I told you, single binding site, that is affinity, multiple binding sites is avidity. Those who have not attended, otherwise it will take longer time. This I have explained, antigen binding site, this is papain cleavage site, PAPA-N, duto fragment. This is pepsin binding site. You get two fragments. See, papain digestion, papain, এই যে দুটো ফ্র্যাগমেন্ট পেয়ে গেলে এখানে একটা ফ্র্যাগমেন্ট পেলে, ঠিক আছে? তাহলে আমরা এই অ্যান্টিবডি বাইন্ডিংটা ইউজ করতে পারি, একটা ড্রাগের সাথে ট্যাগ করাতে পারি। We can use this one, and we can, this part is having carbohydrate molecule attached to a protein molecule, this is antigenic, this part we can remove it, and we can use where there is a complement is required, activation is required. So pepsin digestion will cause FAB2 fragments. You get my point, what I have explained? So in the simplified way, this is again, these two fragments what I have written. This it is available in the book, you concentrate, you read it, you will remember. So classification, as I told you, there are 5 chains. Now you look at this molecular weight. The IgG is having a molecular weight of 150 because of the composition of the amino acids. Now it is IgA is a monomer, but its molecular weight is ranging 170 to 400 because of the amino acid composition. IgM, so 150 into, just roughly, 150 into 5, that becomes 750. 150 multiplied by 5. So that will be 700, approximately 750, and on top of which you will get some, because of the J chain is there, that is having some molecular weight. So that is the reason why its molecular weight is 900. It can be IgG, 5 IgG molecule, it is 150 means may not be, every case may not be 150. 150, 151, 149, 150.12, because of the amino acid composition, roughly. IgE again a monomer, but its molecular weight is 190. So can you see that IgM is, since it is a pentamer, it is having a structure of this 900. IgA is a dimer. So that's why 150 plus 150, that is 300, on top of which you will get that J chain. So maximum concentration of IgG is present here. IgG is present in the highest concentration, remember. It is in the blood. IgA is present mainly on the mucosal surface. IgM is responsible for primary response. IgD is present in some receptors also, in the IgE specifically for allergy. I will give you the chart. So again, immunoglobulin subclass, if you go, there are 4 types of subclasses. Again, IgG1 is 66%. Similarly, if we go subclassification, the IgA1 and IgA2, note down this one. These 4 parts are written, this you may not get. But anyway, this part you write down, this chart. Primary response, this Ig, what is the difference between the 3? One is IgM, IgG and IgA. What is primary response is IgM, as I have repeatedly told, you will get it. But IgG, IgA will not be there in the primary response. External secretions. Remember, external secretion IgA. So IgA is present. Placental transfer. From the mother we get IgG. Agglutination. IgM is a strong agglutinating antibody because they are having 10 binding sites. So this IgG can also cause. IgA having 4 binding sites, 2, so therefore it is having a double plus. Complement fixation, you may not remember. Your Ig is related to allergy. Anybody suffering from allergy, usually Ig levels in the blood is asked to get what is the level of IgE. If the level is high, it is matching with the anti-allergy reaction, and it is secreted by the mast cells. 3 different types of antibody is there. One is, note down this one, isotype, allotype and idiotype. What is isotype? Means these 5 chains, what I have said, 5 different chains are there. IgM, IgD, IgD, IgA, these 5 types are called, these are called isotype. Allotype, that in the heavy chain there will be difference. Suppose I am having an antibody, you are also, all of you are having antibody. So all the antibody IgM type or IgG, IgA will be there, but it will not be same. It will not be identical. So there will be some variation. So what we are having, African people may not have, American people may not have. So these are studied for population genetics, population study. And allotype, suppose if I am suffering from bone marrow cancer cell, bone marrow cancer. Now my healthy cells are somehow sequestrated, but separated. Now they are grown, they are transfused on to me again. That is called an autologous graft. Now this cannot be done in my case. I require somebody to donate for me from the same species. It is called allogenic. So allotype, now you understand, you are having a different type of antibody, same thing, but there is structural difference. And idiotype, it is basically in the hypervariable region, because we develop antibody against different types of antigens. My idiotypic variations may not match with you. By chance it can match with him. They are having the same antigen, both have been exposed, both have developed antibody. So that minor structure difference in that variable region is called idiotype. So allotype variation is in the heavy chain region, idiotype region is in a variable chain region, and isotype is the main 5 classification of the heavy chain. Just I am giving you an example, isotype. Now you see the look at this structure. This structure is black, this structure is somewhat sky blue, this is somewhat, I mean, having another different color. So the heavy chain difference, the chain is altogether different heavy chain. So that is what is isotype. Allotype, there is a difference in the heavy chain, but not in the variable region. Constant region there is a variation. That is allotype. And idiotype is on the variable region, not on the constant region. এই চারটি লেখো, এইটি তোমাকে লিখতে হবে আর এটি মুখস্থ করতে হবে। Gamma, I try to understand. IgG is antibody of secondary response. Note down red points at least. Old exposure of infection. If you get IgG of dengue, that means she has been previously exposed to dengue. It is not a new infection. And that is how you diagnose a case of fever. Whether he is suffering from recent dengue or a past dengue is also there. Same typhoid. So only class transported across placenta, as I told you. Opsonization of microbes, I told that antibody is having a different half-lives. That's why there is, after primary response, there is a decline. Again in the secondary response you will get, and there is a height and there is some decline. Because the antibodies are having some half-lives, and the half-lives also vary. IgA, repeatedly I told it is present in the, gives a surface immunity. Sorry. So primary defense at the mucosal membrane against local infection. Suppose you have taken into a bacteria, virus, anything, whatever you have taken, so the mucosal immunity will not allow that particular pathogen to enter into our system. So half-life is 6 to 8 days. Delta is present in all certain lymphocytes. IgE, remember, major antibody for allergy, I repeatedly told, and anaphylaxis. And greatly increased in allergic reaction, I told. IgA mu, it is a primary response, difference. IgM primary response, IgG secondary response, IgG and other types, predominantly IgG is secondary response. And as I told you, high agglutinating capacity, I have explained why it is high agglutinating, and complementary infectivity can be produced by fetus, it can, but undergoing an infection. See why we are able to produce so many, this is one important part, only understand. This picture you might get in the book, but now understand this part. You need not write down. What you have to write down, I will give one example, that picture, next picture you note down. So we are having a million of antibody in our system. How? We are having 150 genes, but there are 1 million antibody. This is written in Harrison's book. I have checked with DM Vasudevan, that is written 10 to the power 10, that comes about 1000 crore. And yesterday I checked Wikipedia also, that gives a 3 into 10 to the power 11, means 30,000 crore different types of antibody. So how these different types of antibodies are produced? It is because of, write down in the VDJ joining of heavy chain, write down. Basis of antibody diversity is because of VD and J joining, or VDJ joining of heavy chain and VJ joining of light chain. And how this happens, I am telling you. VDJ joining of heavy chain, তিনটে জিনিস, আর VJ joining of light chain. এই জায়গাটা দেখো, তাকাও ভালো করে। এই this part is V, this part is D, this part is joining J chain and this part is constant. In the heavy chain, in the light chain we are having B and J joining, nothing more than that, B and J joining. I am explaining. This structure you write down, this image you write down, you will not get it. How? Be a little faster, or better you can take a, right now I am allowing you to take a photograph. You take a photograph to save time. You understand this part, then you match with this thing. Nina photo Nina nicho ever explain bojache bojo ei part ta. Suppose ekhane dekho why diversity, how this VDJ joining occurs. Suppose there are 4 V regions. We are having several locuses, we are having 150 genes. So J is having, suppose there is 4 D is there and 4 J is there. Constant region also is having different things, but I am only allowing to understand VDJ joining. So we are now removing this part, this 2D and 2J we are removing for what creation of one antibody. So removal of unwanted D and G gene, এই দেখো D টা কাটছি এবং G টা কাটলাম, ঠিক আছে? এই কাট পার্টটা যদি কেটে দেই, if this part is deleted, now what structure we will get? We have not touched this part, variable part we have not touched yet. We have part only D part and J part. So we have cut it, so we got 4V, but this 2D is there and 1J is there. Okay? So this 2D is there, 1J is there. So this DJ combination will be occurring. Now DJ combination is there. Got it? Now in the next step, we don't want this thing. V, we don't want this. V and D. This part we don't want. Now we are deleting it. So again V and D segment we are removing. So at the end what you will get? এই দুটো V পেলে, D পেলে, J পেলে আর C পেলে. Now you got it. So by doing that thing, we get an innumerable number of VDJ recombination. Same thing happens, suppose if there are, it's a nature communication I have from the latest reference I got it. So there are 129 V variable region, 27 diversity and 9 G chains. Only write down one part, you have understood that thing? Now write down this upper sentence. Somatic recombination of multiple germline gene segments due to exposure of different antigens in individuals given time. লেখা না এটা লেখো গোটাটা লেখার দরকার নেই, লেখো তো যেটা ছবি তুলতে দেবো বলে দেবো, এটা বুঝে নিয়েছো তো ব্যাপারটা। So this change today it has been covered, suppose it is July 23 reference I am telling you. Now after July 33, this number might increase, this number will be increased, there will be alteration of numbers. So just basically understanding that we have to get multiple copies and we will get a combination of the recombination of the two, permutation simple. So for one antigen we got a one combination, for another antigen we will get another combination. So same thing it is again taken from your Vasudevan book so that you understand when you go, it is given for light chain, not for heavy chain. So light chain VJ joining is there. Suppose here it is there, what is in? The VL is 500, J is 6 and C region is 20th. Suppose we are taking the 20th position here. The same thing, that picture what you have taken in the photographs, 20th we have taken this one, J chain we have taken this one and CL is there. So you, at the end you will get a gene like this and that gene will be transcribed. So there will be matching, definitely there must be matching with the light chain and heavy chain, otherwise that will not be able to bind. So diversity basis is basically of the heavy chain, the diversity region. Do you get my point? So because of that thing, again variable region, again Harrison's medicine, there is this what Dr. Vasudevan book has written. I have checked it whether it is the same figure, because in the earlier editions also the same figure is there, they simply copied. But now, because since you will be reading medicine, deliberately I am taking you from medicine book. So the Harrison latest book is available in 2023, that is Kappa-Chen is 370. Why I am telling you? Because there will be several confusions between different books. The best reference book will be basically for you, Harrison, a medicine book. Nobody can challenge. Whatever book Indian authors you are telling, this is a reference book. And if you, the moment you quote that this again I am telling you, this 150 genes, IG genes and 1 million antibodies, this is in a Harrison book. So basically some of the information is deliberately I am giving you from clinical books, because if you remember that thing, that will be helpful for your clinical years. So that Susumu Tanaka name might be seen, she got a Nobel Prize because of this antibody diversity, because there is no clue why so many antibodies are how produced. That VDJ combination, recombination is there, and that's why he was. So antigen antibody binding is just like protein binding. So there are three forces you remember, is electrostatic bond, van der Waals force, hydrogen bond. This is required for protein binding, this is required for enzyme substrate binding also. So what are these things you read very clearly, so that you will be able to understand. A basis of autoimmunity and autoimmune disease is failure of recognition. If you attack one system, then it is called autoimmune disease. And most of the autoimmune diseases are having a psychological background. That though it is not written in other books also. And I have seen it during my genetics course. That most of the autoimmune diseases are having some amount of psychological component. So the moment you take care of the psychological component, autoimmune disease gets cured, and that is a fluctuation, because stress and strain doesn't remain same. So you if you get an autoimmune case in your clinical practice, you try to spend a little time to understand the psychology. Perhaps you will be able to manage the case better than others. So common autoimmune diseases are there. Coming to main part, disorders of immunoglobulin, that part you just, I will take 10 more minutes. See, plasma cell, this part is required, because I cannot cover it, because I went slowly, very slowly. This is one part you will be asked during your examination, that is M protein, that is the thrust area. I will explain what is M protein and why it is called like this. So basically B cells are, the immunoglobulins are produced for the B cells, isn't it? So what will happen in the disorder? Either the B cells may not function, that plasma cells, there will be hyperfunctioning, less function or no function. If there is less function, then there will be hypoglobulinemia. If there is no function, there is aglobulinemia. That is as simple as this. Or hyperfunction. If it starts producing hyperfunctioning, like autoimmune disease, we have developed antigen against our antibody against our own antigen. So antibody will be produced on level, if you say that will be higher. So liver disease, common liver disease, normally what happens, basically, if you classify the total protein, see, আমরা করি কি total protein মাপি, is total protein and albumin. A TP suppose TP range is 6 to 8 and albumin range is 3.5 to 4.5 grams per deciliter. Simply what we do in the clinical practice, suppose somebody's total protein is 7 and somebody's albumin is 4 albumin. So globulin is basically 3, isn't it? So AG ratio, this is called AG ratio. AG ratio is normally 1 to 1.5 is equal to 1. অর্থাৎ অ্যালবুমিন সবসময় বেশি থাকবে, মোটামুটি মনে রাখবে 1.5 সবসময় অ্যালবুমিনটা বেশি থাকে, 1.5 to 1, 1.5 to 1 is to 1. Albumin is always greater. Albumin level এখানে 4 দেখো, এখানে 3. বাই চান্স এটা যদি রিভার্স হয়ে যায়, সাপোজ অ্যালবুমিন পাওয়া যাচ্ছে 2 আর গ্লোবুলিন পাওয়া গেল 5, হতে তো পারে। দিস ইজ রিভার্সাল অফ AG ratio. The more commonest cause of reversal of AG ratio is abnormal globulin. Plasma protein পড়ানো হয়েছে তো? What are the different types of globulins? Can anybody tell? What are the different types of globulins? Alpha 1, alpha 2, yes? Huh? Alpha 1, alpha 2, correct? Beta, gamma. কোনটা বেশি কনসেন্ট্রেশনে থাকে বলো তো? গামা। তারপরটা ঠিক বলেছে, বলো বলো, চেষ্টা করো, বিটাটা পাবে, তারপরটা আলফা 2 আর তারপরটা গামা। এবার মনে করো একটা মিক্সচার পেয়েছি, আমরা কি করেছি, এটা একটা বুঝিয়ে দিচ্ছি এখন, এই জায়গাটা ভালো করে বুঝে নাও, তাহলে রেস্ট ইউ উইল আন্ডারস্ট্যান্ড দিস থিং। হি হ্যাজ সেড কারেক্টলি, উই গেট অ্যালবুমিন, উই গেট গ্লোবুলিন। এটা হচ্ছে কি কনসেন্ট্রেশন থাকে আলফা 1, আলফা 2, বিটা আর গামা, ঠিক আছে। এইবার মনে করে একটা মিক্সচার, এই মিক্সচারটাকে সেপারেট করতে হবে। দ্যাট ইলেকট্রোফোরেসিস ওয়াজ টোল্ড ইন দ্য প্লাজমা প্রোটিন ক্লাস। This part I have to tell because otherwise you will not understand this thing. Now if we get a mixture of all these things, these are all proteins, how will you differentiate? We cannot differentiate. So what is done basically, each protein is having some pi value, means isolated point, some part of the proteins will be positively charged, some will be negatively charged. So we have to first make this thing, everything is negative. We change the pH so that all proteins becomes negative. Now if we allow this mixture and we place something like suppose in a paper, just or a gel, maybe it's a paper, it's maybe a gel, and normally it is done in agar gel, plasma protein. You try to understand this thing, then everything will be clear. And if you cannot understand, just stop me. We basically, after making a gel, we cut a small portion of that gel. Gel is just like a jelly-like substance. And in that gel, we place this serum, which everything has been made through an alkaline pH, we made everything negative. Now this is an electrical field. Now electrophoresis, I'll explain this thing, this picture you see it. Multiple myeloma is common, just wrote down, and another is this is, I have written basically just to explain this thing, this is not written anywhere. Hyperfunction, disturbed function. Disturbed function means disturbance is there, and it is usually hyperfunction, and it causes in the malignant condition usually, overproduction of antibody, and that plasma cell dyscrasia came into in the second prop examination, full length question paper. So plasma cell, if this undergo disorder or dysregulation, that will produce an abnormal immunoglobulin, high amount of immunoglobulin, and these are all cancer cells basically. So this is MUGS monoclonal globulin, not write down five diseases, only this part. This plasma cell dyscrasia, because this multiple myeloma will come. MUGS monoclonal gammopathy of MGUS হবে এখানে, MUGS না, MGUS of unexplained syndrome, আর একটা যে প্লাজমাসাইটোমা, মাল্টিপল মাইলোমা, এইগুলো বইয়ে পাবে। দাঁড়াও এবার জিনিসটা বুঝিয়ে দিচ্ছি, মাল্টিপল মাইলোমা কেন বলে, মাল্টিপল সাইটস হয় বডিতে। এইবার তাকাও ভালো করে, এই অ্যালবুমিনের কনসেন্ট্রেশন দেখো, জাস্ট লুক অ্যাট দিস থিং, do not note down anything, just understand this thing. So albumin maximum concentration আছে এখানে দেখো, গ্লোবুলিন তারপর আছে আলফা 1, আলফা 2, এই দেখো তুমি যেটা বলছিলে, তাকাও ভালো করে, এই দেখো গামা গ্লোবুলিনটা সবচেয়ে বেশি থাকে, আলফা 1টা কম থাকে, তারপর আলফা 2টা বেশি থাকে, তারপর বিটাটা থাকে, ঠিক আছে। আর এই যে গামা গ্লোবিনের ফ্র্যাকশনটা, এইখানে 5টা ইমিউনোগ্লোবিন থাকে। হ্যাঁ, এই গামা গ্লোবিন ইজ অলমোস্ট ইকুয়েটেড টু ইমিউনোগ্লোবিন। হোয়াই? দ্যার ইজ ডিফারেন্স। গামা গ্লোবিন কী কারণে হয়, কেন তোমাকে হচ্ছে, এখনই বোঝাচ্ছি এখনই। আর ইমিউনোগ্লোবিনটা বেসিক্যালি এই ক্লাসে কেন পড়ে, কারণ 5টা ইমিউনোগ্লোবিন এই জায়গাটাতে আছে। এই দেখো, এই তাকাও, এবার এই জায়গাটা বোঝার চেষ্টা করো, এই কনসেন্ট্রেশনের দিকে তাকাও ভালো করে। এবার এখানে আমি এটা প্লেস করেছি, নর্মাল লোক, নর্মাল, উই আর অল নর্মাল, উই হ্যাভ প্লেসড অ্যান্টিজেন। এইবারে এই মিক্সচারটা তৈরি হয়ে গেছে। এর মধ্যে সবচেয়ে লো মলিকুলার ওয়েট আছে অ্যালবুমিন, 67,000-এর কাছাকাছি। তো এবারে ইলেকট্রিক্যাল ফিল্ড আমরা অ্যাপ্লাই করি কোনোভাবে এখানে, এটা একটা ফিল্টার পেপার নিয়েছি ওখানে, বা ফিল্টার পেপার বা জেলটা নিলাম, ইলেকট্রিক্যাল সিস্টেমের মধ্যে ঢোকালাম, এবারে ইলেকট্রিক্যাল কারেন্ট দেওয়া হচ্ছে। এটা একটা নেগেটিভ, এটা পজিটিভ সাইড আছে। তাহলে যখনই আমরা এখানে নেগেটিভ চার্জ, যেগুলো সব করে নেগেটিভ চার্জ, এখানে যে ইলেকট্রিক্যাল ফিল্ডে দেবো, নেগেটিভ থেকে পজিটিভ দিকে মুভ করবে। তো পজিটিভ ইজ অ্যানোড, দিস ইজ ক্যাথোড। যার মলিকুলার ওয়েট সবচেয়ে ছোট, which having smallest, it will move fastest, and so albumin will move fast. তারপরে দেখো বিটাটা আসবে, তারপর আবার ওই দেখো, তাকাও ওইদিকে, এটা দিকে, আই দেখো, ওই দেখো অ্যালবুমিনটা এসছে, তারপরে এই একটা ব্যান্ড আসবে বিটা, তারপরে এই আলফা 1, তারপরে আলফা 2, বিটা, তারপরে গামা। যেহেতু অ্যালবুমিনের কনসেন্ট্রেশনটা খুব বেশি থাকে, সেই জন্য অ্যালবুমিনটা এরকম থাকবে, একদম পুরো একদম শার্প একটা ডিপ পয়েন্ট নেবে, বুঝেছো ব্যাপারটা? তারপরে দেখো এইটা ছোট্ট মতো নেবে, কারণ এর কনসেন্ট্রেশনটা তারপরে আছে। তাকাও ওইদিকে, তাকাও, অ্যালবুমিনের কনসেন্ট্রেশনটা আলফা 1, দেখতে পেয়েছো? দিস ইজ অ্যালবুমিন। তাহলে এইটা যদি হয় তার চেয়ে একটু বেশি পাবে এইটা, এইটা আলফা 1, এটা আলফা 2, এইটা বিটা। আর গামাটা এদের চেয়ে একটু বেশি পাবে, একটু হেটেরোজিনাস পাবে, কারণ কি মলিকুলার ওয়েটটা অনেকটা ডিফারেন্ট আছে। দেখলাম না, 150 থেকে প্রায় 900। তো কেন এই যে শার্প পয়েন্টটা রয়েছে, এটা দেখো কত ডিপ কালার নিয়েছে, দেখতে পেয়েছো? তারপর দেখো আস্তে আস্তে কালারগুলো বাড়ছে, আর গামা রিজনটা একটুখানি হেজি হয়ে গেছে, কারণ এখানে মলিকুলার ওয়েটটা ডিফারেন্ট আছে। এইবার কোনো কারণে যদি ক্যান্সার হয়, কোনো একটা ক্যান্সার হয়েছে, মনে করো সাপোজ প্লাজমা সেলের টিউমার হয়েছে বেসিক্যালি, তো আইজিজি, আইজিএম, আইজিডি, যা খুঁজে একটা অ্যাবনর্মাল ইমিউনোগ্লোবিন তৈরি করবে। Abnormal immunoglobulin produced by that plasma cell is called a paraprotein. আর ও তখন করবে কি, এই রিজনে এই একটা বিরাট অত্যন্ত ডিপ একটা ব্যান্ড তৈরি করে দেবে, and that is what is called M protein. এইটা গামা পজিশনে থাকতে পারে যদি মলিকুলার ওয়েটটা বেশি থাকে, এটা বিটা পজিশনে থাকতে পারে। মলিকুলার ওয়েটটা যদি 6 থাকে, ক্যান্সার সেলের তো কোনো ক্যারেক্টার নেই গো, তো ইট ক্যান প্রডিউস লোয়ার মলিকুলার প্রোটিন অলসো। সো when it is present in the gamma position, that is called gamma myeloma. When it is present in the beta position, that is called beta myeloma. And this abnormal protein of multiple myeloma is called M protein or M band. This is called an M band. Do you get my point? The abnormal amount of globulin will be produced that may be exceeding the amount of albumin also. A cancer, advanced cancer, too many plasma cells are there. So the thickness of the antibody band from this electrophoresis pattern, you will be able to diagnose in the OPD itself. Show me the report. Do you get my point? So this is what is the importance of immunoglobulin. Now if you want to basically actually measure how much is there, this is apparently you are seeing, now you have to, during your practical class you will be doing colorimetric exercise. There you will quantify what is the exact amount. So then for densitometric scanning is that a graph. This graph is basically densitometric scanning. We use a scanner. What is the concentration here? You see the concentration is very high. That's why the peak is very high here. Now the concentration is, the peak is here and gamma is just like that. But if you get a M band, so this M band will be just like that albumin. Is it clear? Now the gamma and immunoglobulin, gamma is present, most of the immunoglobulins are present in this gamma region. That's why it is called a gamma globulin, and immunoglobulin belongs to this gamma portion of the electrophoresis. So that's why it is called a gamma globulin. So why immunoglobulins are belong in gamma globulin? Now do you get my point? So alpha globulin is here, beta is here, gamma is here. Because of the electrophoretic mobility at the gamma position, that is called a gamma protein. So this M protein, since it is present in this gamma region, it is called gammopathy. So electrophoresis is the same thing I told you. It is moving from negative to positive charge. Filter paper, there are so many fragments will be produced. This picture you will get in your book. Peak. এই বেজোর প্রোটিন ইচ্ছা করে তোমাদের দেখালাম যে রিভার্সাল রেশিও যদি হয় সঙ্গে সঙ্গে ডাউট করবে, যেই মুহূর্তে যদি এই যে রিভার্সাল কোনো কারণে হয়ে যায়, ফার্স্ট ডাউট করবে কেন অ্যাবনর্মাল গ্লোবুলিন আছে কিনা, অ্যান্ড ইন মোস্ট কেসেস উইল গেট এ ক্যান্সার কেস, মাল্টিপল মাইলোমা অর লিভার ডিজিজ। এই দেখো, তাকিয়েছো, দেখতে পাচ্ছ? এই দেখো এম ব্যান্ডটা এইখানে দেখো, এই অ্যালবুমিন, এইখানে দেখো এম ব্যান্ড, M1 ব্যান্ড, এই দুটো পরপর ব্যান্ড তৈরি হয়ে গেছে। এই দেখো এম ব্যান্ড বিটা পজিশনে এসছে, তা বিটা মাইলোমা হতে পারে, গামা মাইলোমা হতে পারে। বোঝা গেল ব্যাপারটা? এগুলো বইয়ে পেয়ে যাবে, এগুলো যেটা বইয়ে পাবে না সেগুলো তোমাদের লিখতে বলি, এগুলো বইয়ে পেয়ে যাবে। বইয়ে পাবে আননেসেসারি লিখে না গো। দেখতে পাচ্ছ স্পাইকটা, তোমাকে বললাম এই যে অ্যালবুমিন, দেখো অ্যালবুমিনের পিকটা নিয়েছে এখানে, অ্যালবুমিনের পিক দেখো সমান সেম পিকটা এখানে আছে, এতটা হাই কনসেন্ট্রেশন। সো ইউ গেট এ densitometric scanning, you get a big peak. So just by looking at that electrophoresis plate in the OPD, you will be able to diagnose. So multiple myeloma, again this thing, this band I am telling you, this is the band of that black band. Now I think you have understood this thing. So this M band, this you will get, I am telling you, this will allow, don't waste time, you try to understand this part. So the monoclonal proliferation is called, that will produce an antibody response. Now polyclonal can occur if there is a chronic infection. There will be polyclonal, means multiple clone of cells will be involved. So you will get a hazy portion here, beta gamma combination. You will get the heterogeneous type of things. So this beta gamma combination will be there. Now I think you read this thing in the book. Benzone protein, one line, you have been told in the urine class, I think. In the urine class, this has been told, benzone protein. Are you clear about it, or should I explain? Benzone protein, nothing is found in the multiple myeloma. In only a number of cases, I have not seen a single case in my 30 years career of benzone protein, although I have seen multiple myeloma cases, nearly 20 cases, because this is not detectable. And that's why, what is the characteristic feature? Now, if you, I am telling you, if you take a albumin solution, you have done it, you have heated it, you found the coagulation, and this coagulation will not dissolve by any means. That is an irreversible denaturation. Now, benzone protein, what is the characteristic is that if you heat it to nearly 50 degrees centigrade, 40 degrees centigrade, there will be coagulation, and then heat it higher, it will redissolve. And if you cool it again to this position, that will be again precipitating at 60 degrees centigrade, and if you bring it down to 40 degrees, again it will dissolve. That is the difference between the benzone protein and this thing, normal albumin. So this In your clinical practice, I am telling you, advising you, don't go for Benz Jones protein, you will not get it. Advise the patient to go for, this you note down, this is not written, immunofixation electrophoresis of urine. Just go directly, you will get it. If the light chains are excreted in cancer, this is a light chain, Benz Jones protein is basically light chain. So in urine, if you try to do that thing, you will not get a positive result. And you might miss. But this I have learned, honestly speaking. In 2019, I have attended an Asia Pacific International Conference, and there I raised this issue, and somebody told that you go for immunoelectrophoresis. Immunofixation electrophoresis, you will get it. And I have attended that lecture. So that is the purpose of going somewhere else. You can interact, you can learn something. I raised my concern that a 30 years career I could not get it. Okay. So this understand and you can better. Now this part is clear, this electrophoresis part. বলো লিভার সিরোসিস কেন হয় বলেছি লিভার সিরোসিস শুনো না লিভার সিরোসিস নেফ্রোটিক সিন্ড্রোমে কি কারণে হয় বলতে লিভার সিরোসিস হলে হয় কি শুনে না ভালো করে জাস্ট ট্রাই টু আন্ডারস্ট্যান্ড all the plasma proteins are produced by the liver except globulin. Globulin is synthesized by the B cells. Remember this thing. So any liver disorder, if there is a chronic liver disorder, the liver chronicity, we do not know the nature of the disease. So all the albumins will be, albumin is a predominant globulin, so that will be decreased. So lowering of albumin, hypoalbuminemia, I repeat, I am telling you once again, this will be asked. Two causes of hypoalbuminemia, one is, first cause is nephrotic syndrome. It is getting leaked, albumin is leaked through the urine so that you get a hypoalbuminemia. And second is decreased production by the liver. Liver is the main site for albumin synthesis. So when the disease occurs, this is particularly this autoimmune disease affect the liver and they chronically affect the liver disease. Since it is autoimmune disease, so antibody response will be mounted, so antibody production will be there. In addition, the albumin level will be decreased, so you will get an AG ratio reversal. So because of the heterogeneity of that antibody and the molecular weight, you will get a beta gamma region because abnormal production will be there. Cells are no longer producing the normal things because the liver is now diseased. And the commonest cause of beta gamma albumin, I am telling you, if the moment you get an AG ratio reversal, in an otherwise normal individual, you think of two things. One is liver disease, chronic liver disease, just palpate the liver or go on ultrasonography, or you think of cancer, this might be a case of multiple myeloma. Invariably you get out of, at least you rule out these two things and then you proceed for other things. AG ratio reversal, commonest cause is if it is a too high, remember, if it is a too high AG ratio, albumin is 1 or 2 and globulin is 8, 10, that means go for it, it's a multiple myeloma. If it is something like that, less, suppose albumin level is 3 or 2 or 2.5 and globulin level is 4.5, think of some chronic liver disease and other chronic infections. Since liver disease is quite common, we think of chronic disease or chronic infection is there. Today in our system, except in the a little bit affluent classes, most of the people do not suffer from that chronic infection. It is common prevalent in the poorer sections of the society. Okay, this part is clear. Why beta gamma region, you know, understand? Because chronic liver diseases are usually autoimmune diseases. So they mount an autoimmune response against own body tissue. And that is the reason globulin levels are increased. In addition, because of disturbed functions of other albumins, other plasma proteins, you will get abnormal levels of plasma proteins also. So in that case, you ideally you go for electrophoresis and you will get a broad band. Anything else?